Enzymes are catalysts for reactions, which means they help speed up chemical reactions. Enzymes are proteins made up of amino acids. They are found in the body and help with digestion and metabolism. A substrate is a chemical species that is observed in an experiment. The name of an enzyme is usually derived from its substrate, with the word ending in ase.
In our previous lab, we tested the number of substrates disassembled using 2 enzymes over the course of 1.5 minutes, using various amounts of substrates to learn the results. We wanted to take that model lab and test a real reaction. In class, we did an experiment where we tested the effects of using an enzyme to break apart substrates versus not using an enzyme. We took our own spin on the lab to test the amount of oxygen released with different substrate concentrations. We performed 4 trials using 4 different hydrogen peroxide concentrations to test how much oxygen would be released with each concentration.
Peroxide can be referred to as 2H2O2, which is its chemical sequence. The peroxide is then broken down into 2 parts, 2H2O and O2, and that is where the oxygen that is being released comes from. The 4 different concentrations of hydrogen peroxide, or H2O2, that we used were 6%, 3%, 1.5%, and 0.75%. We mixed 10 milliliters of each H2O concentration with 1 milliliter of our enzyme peroxidase in each trial. We put this solution into a closed reaction chamber that contained a tube in the lid which traveled into the inverted graduated cylinder filled with water. The oxygen produced from the enzyme substrate complex traveled through the tube into the graduated cylinder where we measured and recorded the results after 5 minutes.
The first trial we tested was with 6% hydrogen peroxide. The enzymes were breaking down a lot of substrates, like a lot. This concentration only released 9 milliliters of oxygen. This happened because many of the enzymes were denatured since there was too much pressure from all of the substrates.
In our second trial, we tested a 3% hydrogen peroxide concentration. The amount of oxygen grew rapidly at the beginning of this experiment. Overall, this concentration released 30 milliliters of oxygen, but during the experiment, the enzymes were getting more and more denatured, resulting in less oxygen being produced, but some were able to keep going.
In our third trial, we tested a 1.5% concentration. Throughout the experiment, there wasn't as drastic of a decrease in oxygen production as there was in other trials. This shows how little the substrates denatured the enzymes, meaning that there weren't too many substrates per enzyme, creating the most success in releasing oxygen. This was our best trial in terms of how much oxygen was released with 37.5 milliliters of oxygen.
In our final trial, we tested a peroxide concentration of 0.75%. In the beginning, they had a pretty rapid start, but it began to decrease drastically. There was only 24 milliliters of oxygen being released. This is because there were too many enzymes for the substrate. As you can see in our graph, the hydrogen peroxide concentration that released the most oxygen was the 1.5, followed by the 3%, and then the 0.75%, then the 6%.
In this model, you can see the reaction chamber and the tub of water with the inverted graduated cylinder and the tube flowing into it. In the reaction chamber, the big black Pac-Man-shaped figure represents our enzyme peroxidase. A couple of the enzymes are denatured, which is shown by the squiggles. The brown triangles are H2O and the purple triangles are O2, and together they make our substrate hydrogen peroxide. When the substrate connects with the enzyme, the H2O and the O2 are split apart. The H2O sinks to the bottom of the chamber, and the O2 travels through the tube into the graduated cylinder. The oxygen accumulates from there. This model represents the 6% hydrogen peroxide. This percentage releases the least amount of oxygen, meaning there are more substrates left in the reaction chamber. Here, the blue triangles represent the O2. The 3% released more oxygen than the 6%, so we showed less substrate pairs in the reaction chamber. We also showed more denatured enzymes in this trial. Here the pink triangles represent the O2. The 1.5% released the most oxygen into the graduated cylinder. We have the least amount of denatured enzymes and more H2O left behind. Here the orange triangles represent the O2. The last trial was the 0.75% with the second to least amount of oxygen released. This one had another denatured enzyme.
Before doing this lab, we didn't understand the effects that the substance dilution would have on the amount of oxygen released. We originally made the prediction that the higher percentage of peroxide, the more oxygen that would be released. And that was certainly not the case. After this experiment, we now understand the higher peroxide only means more denaturing. Therefore, less oxygen is released. Our group did really well on coming up with an idea and a concept that we wanted to do. Nothing really went wrong. Our experiment relates back to the bobtail squid Vibrio fishery because within this bacteria there is a chemical reaction between the enzyme luciferase and the substrate luciferin. Light is released during the oxidation of luciferase. Luciferase contains two proteins, Lux A and Lux B. Luciferase comes in contact with luciferin, calcium, adenosine triphosphate, or ATP, to produce light.